Welcome everyone to the STEM Session 9 talk of the 6th Polygen Symposium of Rising Scholars. My name is Andrea Joseph from the University of Pennsylvania, and I will be moderating today's talk. The goal of the symposium is to celebrate and showcase the hard work of our Polygen scholars, all of whom have worked tirelessly on their projects over the past couple of months. Today, we'll have 4 student presenters presenting on a variety of topics. A fellow Polygence mentor will be scoring each student's presentation, which will help us determine prize winners after the event. Please type your questions in the chat if you have any for each speaker. We will allot 2 minutes for questions after each presentation. Today's speakers are Pratham, Samantha, Aneesh, and Baran. So we can get started with Pratham's presentation.
All right, perfect. I'm just going to share my screen. All right. 4 million athletes experience concussions annually. In fact, according to a study from the University of Pittsburgh, more than 300,000 of these traumatic brain injuries stem from American football. Inherently a violent sport, football is known for its constant savage tackles and brutal helmet-to-helmet collisions. But how can we stop this and the accompanied brain degeneration, or CTE? Hi, I'm Pratham Chhabra, and I'm here to solve the problem. Today, I will be walking you through what exactly is chronic traumatic encephalopathy, or CTE, the impact of them on the sport, the problems with concussion in football, and a comprehensive 3-step method to solve this critical issue. Next, I will look to discuss my research through my findings and through the insights I extracted from my analysis process. And finally, I will be honored to answer any of your questions. CTE, a form of brain degeneration, is caused by repeated brain traumas or head injuries such as concussions. Despite the use of helmets, due to repeated impact, CTE is quite prevalent among the sport's most idolized stars. In fact, the United States National Library of Medicine explains that CTE causes a progressive decline in memory, cognition, and contributes to depression, suicidal behavior, poor impulse control, and eventual risk for diseases such as Parkinson's and dementia. The immediate impact of such injuries does not go unnoticed. Although unlike a progressive decline in the protein of the brain, headaches, confusions, a lack of coordination, muscle loss, and dizziness, even excessive fatigue, are all potential symptoms because of concussions. With helmet-to-helmet contact occurring with such an astronomical tendency, numerous concussions actually go undetected or are not properly addressed by medical officials, threatening to induce CTE later in these athletes' lives. In fact, head injury experts explain that 95 G-forces, 1 G-force representing the standard gravitational force on Earth, are enough to suffice for a concussion. And believe it or not, the average football tackle or hit exerts 105 G-forces of strength. Simply take a look at the emotional and physical toll the sport takes on its players through concussions. That's truly saddening. Many of the complications associated with concussions and progressive brain degeneration are preventable, but currently, what even are the problems? Well, countless of these injuries are a result of inefficacious helmet technology. None are engineered for concussion elimination. Secondly, safety is simply not a concern. Ultimately, many of these injuries stem from dirty or unsolicited hits, inadequate training, or even ineffective gear. Per health organizations worldwide, these injuries can be prevented. However, it is evident that proper equipment guidelines and training must be integrated into the game in order to do so. In order to analyze chronic traumatic encephalopathy risk in American football players without actually viewing brain scans, I contacted the National Football League's very own Health and Safety Department and was given access to their database of records. Spanning from 2012, we used a 3-year sample of data until 2015 to conduct our research. As such, we use Python's data filtration capabilities to parse through over 10,000 individual injury records. First, we use the pandas data frame or package to refine our statistics to strictly concussions, a form of brain trauma. This allows for maximum alignment to the brain degeneration insights that we saw. Subsequently, we retrieved each athlete's name, position on the field, association to a team, total games missed, and finally, what season this report is taken from. In doing so, we reviewed a comprehensive numerical data manipulation process, and we had the appropriate setup for an organized data sheet. With the respective year, total concussions, total games missed, and positional groups listed, the organization allowed for the most efficient process with over 16 columns in organization. Well, what did I concur through my research? First, I made crucial chronic traumatic encephalopathy predictions in various football positional groups. Predictions on the wins above replacement, or WAR metric, to determine the impact of unhealthy players on a team, and finally, a helmet to prevent concussions that I rendered myself. Through my data analysis process, as can be seen, I uncovered crucial data. Quarterbacks showed 96% more signs of affecting team win percentage more significantly than another positional group. On the other hand, secondary or cornerbacks and safeties accounted for 29.4% of brain traumas. My biomechanical simulation demonstrates the astronomical G-force threshold that is showed. In fact, per research, 99.05% of National Football League athletes are at risk for chronic traumatic encephalopathy and brain degenerative dementia, signaling cognitive dysfunction. Introducing football's first helmet to eliminate concussions. Through working with a sports analytic majoring student at the University of Syracuse to discuss the essential components of a helmet designed for durability in American football, I researched CTE technology that can be implemented into headgear. As can be seen with the engineering structure, I used polycarbonate, ABS thermoplastic, a polymer, a G-force sensor, intelligent energy control cells, and a precision fit lining layer. In fact, the G-force sensor extracts real-time insights to analyze the magnitude of a force to a helmet. Working with the energy control cells, they compress or shear to absorb energy at the impact site. Through using university-level biomechanical simulations of the helmet in action to test impact absorption of the weight exerted onto the frame, the helmet is safe, effective, and most importantly, revolutionary. Now you may be asking, how can all this be applied to the game? Through developing a helmet specifically designed to eliminate concussions, the risk of brain disease in football players is sure to decrease dramatically. With that, we look to redefine what player health and safety means, ensuring a better game moving forward. Thank you. Now, I would be honored to take any of your questions.
Thank you very much, Pratham. As a reminder to attendees in the audience, please feel free to put your questions in the chat, or we can also check the Q&A box. While everyone is, I'll give everyone a moment to type so I can ask the first question. I was interested in what your hypothesis was entering this project in terms of what positions you thought might have had, the highest risk for concussions and CTE, and, then were you surprised or not surprised at your results?
Yeah, that's actually a great question. So I actually was surprised by my results because my hypothesis was the fact that quarterbacks would actually be the most affected, because on TV, I see them experience the most helmet-to-helmet concussions, and it's sort of glorified by the media. But in the real fact of matter through the data, it can be seen that it's more of skill positions such as cornerbacks or secondaries that are being more effective. And this needs more awareness for that.
Yeah, that's super interesting. I see Chris has his hand raised. I think I will click allow to talk.
There we go.
Cool.
Yeah. What was kind of your initial interest in the topic?
Yeah. So actually, since I've been about 5 years old, I've always been hooked to the TV, watching my favorite sports stars, and especially football with the San Francisco 49ers. So I was always super disappointed when my favorite players weren't on the field to play, and even when I was at the games. So I felt like there was always a reason for that. And I realized that it was actually concussions and this brain degeneration that they've been experiencing throughout that have caused them to be sidelined. So I thought, why not take the initiative to make a helmet that can prevent this and actually analyze the root cause of CTE, or chronic traumatic encephalopathy.
Awesome. Thank you, Pratham. I think our 2 minutes of questions are up, so we'll move on to our next speaker, Samantha.
Hi, everyone. Let me go ahead and share my screen. Okay, wait, can you guys see my Zoom screen too, or only the slides?
I can't see your Zoom screen, just
Okay, perfect.
it's not in presenter mode right now.
Yeah.
Perfect. You got it.
Okay, awesome. Hi, everyone. My name is Samantha, and I did my polygens project on the adolescent brain and its relationship to mental health and cannabis use. Hold on 1 second. Okay, there we go. So a quick presentation overview. First, I will be going over my project abstract, and this will cover the question about what I decided to research, and then we'll dive into some research findings about what the data shows. Then we'll look at significance and possible solutions, about why my findings are important and how these issues as far as cannabis use and mental health can be resolved. And finally, we'll look at some conclusions and takeaways about what I got out of this project. So first, we'll go over my project abstract. So obviously, as I said before, the research topic that I decided to look at for my polygens project was the adolescent brain and its relationship to mental health and cannabis use. So some quick statistics and background knowledge that you should have before I dive into my research project is that cannabis culture has been permeating the adolescent population following the liberalization of marijuana, as it is now legalized in 21 states as of now. And a quick statistic that was taken is that 1 in 7 teens report using cannabis within the last month, which is a large increase in comparison to past decades. Also, from birth to approximately age 25, an adolescent's brain is still in development, and this includes supercritical areas such as the cerebellum, the hypothalamus, the thalamus, which are all areas that are super good for homeostasis, and they also help regulate your emotions. So these are obviously very critical areas that are still in development until that age of 25, and they're also very vulnerable to cannabis use and any chemical imbalances. And then lastly, mental health in American teens has been waning, especially with the added stress of COVID-19 and the lack of socialization that many teens have been facing throughout this pandemic. And so the main question that I was really trying to answer during this project was, how does cannabis affect the developing brain and its mental health? So now let's look at some findings. First, I looked at 3 facets when doing this research project: cannabis, mental health, and COVID-19 impact. So for cannabis, I looked at the chemical composition, the popularity within the adolescent population, and then also the long-term effects that are posed by cannabis. For mental health, I looked at COVID-19's impact as well as cannabis impact on mental health and whether that correlation was positive or negative. And then lastly, I looked at COVID-19, its effect on mental health, and also on cannabis frequency as far as usage. So first, let's look at cannabis. One of the main components of cannabis that makes it so addictive is THC. So THC, which is also known as tetrahydrocannabinol, is a neurotransmitter. And essentially what a neurotransmitter is a chemical messenger that helps send signals between other neurons within your body. And this communication occurs throughout the synaptic cleft, which is essentially a large gap between different neurons that allows for communication. And so the THC neurotransmitter also acts as an agonist. And so what an agonist does is it mimics the behavior of other neurotransmitters. And so the neurotransmitter that THC mimics is anandamide. And this neurotransmitter anandamide is very commonly associated with the feelings of bliss and euphoria, which result from cannabis use. And so to carry out these actions, THC binds to the cannabinoid receptors in the brain, known as CB1, and also the ones located in your immune system, known as CB2. And you can see those right here on the graphic as well. And then this increases the rate at which neurotransmitters fire, creating an amplified feeling of that bliss and happiness associated with cannabis use. And then these amplified feelings also partially occur due to the buildup of dopamine in your dopaminergic system. And dopamine also has a lot of associations and connotations with a rewarding behavior. So this rewarding aspect of that buildup of dopamine also this contributes to the habitual nature of cannabis, as it can become very addictive in the future. Some also pointers of cannabis is that it has a lot of psychosocial risk factors when you take it. It can lower your inhibitions, which makes your decision-making and judgments a lot poorer. And then it has a lot of longitudinal issues, such as the reduction of fine motor skills, the declining intelligence quotient, also known as IQ. And then it also increases your likelihood of being diagnosed with a mental health disorder. Okay, next, let's take a look at mental health. So obviously, as I said before, there was a waning mental health problem in the adolescent population. There has been a 5-fold increase in mental health diagnosis to the point where 50% of people, when surveyed that were in the adolescent population, scored in the severe zone as far as their mental health, and also loneliness was considered a public safety issue. And there's a lot of influences that people predict has caused this destruction in mental health, whether that be social media, substance abuse, etc. But no matter what, there has still been an increase in mental health, and that's obviously an issue. And so common diagnoses within the adolescent population include bipolar disorder, major depressive disorder, social anxiety disorder, and general anxiety disorder. And so now we will look at cannabis impact on mental health. So a lot of people, when surveyed in the adolescent population, report that when they take cannabis, it actually improves their pre-existing condition. But contrary to these beliefs, cannabis actually takes a large negative toll on these pre-existing conditions, no matter what it is: bipolar disorder, general anxiety, major depressive. And so, yes, that's what I said before. And so cannabis is a depressant. So no matter if you take it to help your mental health disorder, it will affect it in a negative manner, as there has been seen to be a positive correlation between the two. And then we will look at some COVID-19 impact on mental health and cannabis use. So adolescent cannabis use and mental health issues have only been accelerated by the COVID-19 pandemic. A lot of people who have previously been using nicotine products such as vaping and juuling have been seen to switch over to cannabis products. And also there has been an increase in cannabis use frequency, where people before the pandemic have started smoking cannabis 3.5 times a day. And then from pandemic on, it has increased to 4.5 times a day. So as you can see across the board, there's a positive correlation between COVID-19, adolescent mental health, and cannabis use. And now, as you can see in this graph, 2017, 2018, and 2019, high schoolers had a lower amount of past year marijuana vaping. And then end of 2020, as you can see on the end of this graphic, it has only been seen to increase post-pandemic. So now let's look at significance and possible solutions. So obviously, all of these findings demonstrates the reality of the adolescent drug and mental health crisis in America, and the fact that teens still believe that marijuana use actually improves their mental health conditions further demonstrates how we need to expose these findings to the population that is most vulnerable to cannabis use and mental health, which is the adolescent population. And so then we'll move on to possible solutions. So obviously, there has been a lot of research done in recent years to try to negate this problem. And so one of the breakthroughs was that there's a possibility in controlling the dopamine system through the dopamine receptors, and you can target specific dopamine receptors to combat this addiction. So for example, there's a possibility to manipulate and target specific dopamine receptors, including D1 and D2, which are responsible for the addictive behaviors and can play a key role in solving this issue in the adolescent population. Both the D1 and D2 receptors are involved in withdrawal symptoms as well as relapse behavior. So by manipulating these, it could potentially negate this problem in a really dramatic way. And one of the breakthroughs in pharmaceuticals has been the GSK 598809 drug. And so essentially what this does is it acts as a receptor antagonist, essentially blocking those dopamine receptors to lessen the withdrawal symptoms by creating a buildup of naturally produced dopamine in one's body. And obviously there still needs to be more research to see if there's any possible solutions that we have not yet discovered. So lastly, let's look at some conclusions. So cannabis use and deteriorating mental health has become a dire issue in the adolescent population. And this is very inherent due to the research and the findings that I have previously stated. And also cannabis use, mental health, and COVID-19 factor have seen to have a positive correlation across the board. If one worsens, so does the other, and vice versa, essentially like a domino effect. And then there's obviously an opportunity to rectify the situation in the research in the GSK 598809 drug, and also in manipulating the D1 and D2 receptors. And then all of this data needs to be shared with the adolescent population so that we can try to, in a united front, rectify the situation. Thank you so much for listening. And I would like to give, I don't think she's here, but I'd like to give a huge shout out to my mentor, Kayla, who helped me research and make my literature review for this project. And I'd be happy to answer any questions that any of you guys may have. Thank you.
Thank you so much, Samantha. That was a great presentation. Once again, I can start with a question while we wait for other audience.
Really quick, can I just stop sharing, or do you want me to keep this up?
No, you can go ahead. You can stop.
Perfect. Just so I can see everyone.
Yeah, absolutely. So a reminder to the audience to put questions in the Q&A or the chat. But to start, maybe one conflict that I see is that you mentioned needing to do more research in order to come up with solutions to this problem. But, in order for, I think a lot of scientists would say that it's hard to get approved to do research on cannabis right now because it is not legal in federally or in many states. So what do you think is the appropriate way moving forward? Is it more legalization, or is there another way that you think, or does that have unintended consequences to adolescents, and could that actually make the problem worse, I guess?
So just to clarify, you're asking what steps I think researchers should take in order to do more research?
Is it important to fully legalize weed in order to do
okay.
research, or are the unintended consequences of that potentially worse?
Yeah, obviously it's a double-edged sword, because when you increase the liberalization and the legalization of marijuana, more adolescents will have access to it. And I know for a fact that there are definitely probably a lot of people who are illegally smoking weed right now in whatever states it's legalized in. But obviously that is a step that needs to be taken in order to conduct more research, because the brain is such a complex part of your body. And unless it is legalized, then there's no way of knowing how we can rectify the situation. So I think as of now, since obviously it's not legal in all 50 states, the best route or way of action that you can take as of now would be focusing on the regions where it is legal. So for example, where I live in California, it is legal. So conducting maybe the majority of the research there, because you can have the most participants available to you, would be a good course of action as of now, considering the climate.
Awesome. Thank you, Samantha. Chris has a question in the chat. Do you think your conclusions hold for post-adolescent adults?
So for my research, at least, I focus on 16 to 25 years old. So obviously after your brain is fully developed, it might change a little bit. Granted that when you do start smoking marijuana, or however you may take it during adolescence, those side effects do carry on into your adulthood years. So in some factors, yes, it does carry on into adulthood, because your declining IQ, your lessening of fine motor skills will carry on into adulthood and affect your life then. But as far as things that could affect the brain itself. Obviously, your brain has a lot of plasticity, and so it is able to like rectify that. So I do not have a perfect answer for you, but I will put my email in the chat and I can look more into that and I will let you know.
Thanks, Samantha. I think answered live. Cool. Well, if there are no more questions in the chat or Q&A, well, Chris did raise his hand, but I wonder if that was before.
No, if you have another question, I'd love to answer.
All right, I'll allow Chris to talk.
Yeah, I was just getting tired of typing. You didn't touch on this, so maybe it's not there, but is there a difference between, or was there a variable in your research on cannabis that was consumed via edibles versus smoked?
Explicitly no, but it was focused on like smoking. That was like the research that I conducted when I looked up the main like archive, I guess, where I got most of my information from research papers was from PubMed. And one of the keywords that I did put in was smoking. So it was geared towards smoking, but did I explicitly mention that? No. But yes, it was not through edibles, it was through smoking. Yeah.
Got it.
And if you still want the answer for your previous question, I put my email in the chat. Feel free to message me and I can look more into your question for sure.
Thank you.
Of course.
Awesome. Well, we can move on to our next speaker, who is Aneesh.
Sounds great. Yeah. Can you see my screen?
Yes, I can.
Hi, everyone. So my name is Aneesh Bhat. I am a junior at San Mateo High School, and today I will be talking about the effects of fascia densification and fascia fibrosis on carcinoma. So this topic piqued my interest a few months ago when I learned about statistics related to cancer. So as one may or may not expect, cancer is the second leading cause of death in the United States. And in 2021, the predicted mortality rate of cancer patients was around 32%. So carcinoma, which is a type of cancer in epithelial tissue, accounts for roughly 80% of cancer cases. So understanding the different pathways that may lead to carcinoma could be beneficial, though much research has already been done on that. However, fascia, or more commonly known as connective tissue, has barely been researched in comparison until recently. So now researchers are starting to see a link between connective tissue and cancer, and that's what I'll be going into today. So what is fascia? So fascia, or more commonly known as connective tissue, is a family of collagenous tissue that envelopes and penetrates organs, muscles, vessels, nerves, and joints. So an example I like to use to try to understand what fascia is related to anatomy diagrams. So when looking at different images of systems, not many people think about what is in between that is shown on the anatomy diagrams, and that would be fascia. So its role in the body is critical as it provides structural support and also performs specialized functions for each system. And the tissue is made up of different cells and macromolecules, such as fibroblasts, myofibroblasts, fibrocytes, adipocytes, telecytes, and leukocytes, as well as the macromolecules such as collagen, elastin, actins, and also hyaluronic acid. So one of the most important components of fascia is collagen. And there are several different types of collagen that can be found in connective tissue. And different layers of fascia tissue have different amounts of each type of collagen, as there are many different types of collagen. And these are aligned into specific ways, depending on their location, so that they can perform these specialized functions. So just like many other types of tissue, fascia can become damaged. For example, from limited physical activity, maybe repetitive overworking movements, and also like traumas such as a surgery or even just an injury from like sports. So now going back to collagen, the ratio between collagen type 3 to collagen type 1 is like very crucial to the health and function of the tissue. But when fascia becomes damaged, this ratio of collagen type 3 to collagen type 1 increases. And this is for the worse, resulting in what is known as densification. So densification can later progress to scarring of the tissue, which is called fibrosis, as it makes it harder for layers of fascia to glide over one another. So from either densification or fibrosis, there's an increase in mechanical stress, which can prove to be harmful for numerous reasons. So one of these reasons is the effect it has on the lymphatic system. So the lymphatic system has a significant role in maintaining tissue homeostasis by regulating interstitial fluid and lymph flow. So the lymphatic system relies on two types of forces to induce lymph flow, and those are intrinsic and then extrinsic. So while intrinsic forces come from muscle cells located on the vessels themselves, extrinsic forces come from the transmission of forces from skeletal muscle contractions. And this contributes to around, I think, 1/3 of lymph transport. So connective tissue enables the transfer of forces from the skeletal muscle contractions to the lymphatic vessels. But in order to do so, the fascia must be in a relaxed state. And then if it is, it'll promote lymph flow. However, when fascia becomes dense or fibrotic, as I said before, it is unable to transmit these extrinsic forces to promote lymph flow because of that mechanical stress. So as lymph flow starts to decrease, the surrounding tissue is, I would say, subjected to maybe a slower rate of interstitial fluid recirculation, and this causes inflammation. So the densification or fibrosis of connective tissue, while can lead to inflammation in that way, but can also directly contribute to chronic inflammation. So an inflammatory response occurs to remodel this dysfunctional tissue, which is either dense or fibrotic. And to do so, the migration and presence of inflammatory cells takes place. And this allows fibroblast cells to secrete collagen type 1 and 3 to close these wound gaps. So what this does, though, is that it allows for new collagen bundles to form around the wound gaps, but the new collagen bundles are not aligned with the surrounding collagen bundles, as they were just formed to slowly close this gap. And to change this alignment so that is correct may take years after this initial damage. And while this process is taking place, more stress can occur at the site of injury because collagen bundles are unaligned. And this induces further inflammation and creates a positive feedback loop where inflammation keeps occurring. So inflammation can impact the lymphatic system by inducing contractile dysfunction as well. So as impaired lymphatic function also induces chronic inflammation in the nearby epithelial tissue, what happens is a bidirectional feedback loop between inflammation and lymphatic dysfunction. So why are we talking about inflammation in the first place? It is because this unresolved inflammation taking place in the tissue can create a tumor-promoting environment. So while acute inflammation is necessary as an immune response to combat tissue damage, infection, toxins, and other types of traumas, unregulated chronic inflammation always has a negative impact on the local environment. So when inflammation fails to resolve in the case of chronic inflammation, it is a sign that the initial inflammatory response has failed to heal the site of the damage, or that the normal resolution pathways for inflammation have failed. So this almost always leads to a highly vulnerable environment susceptible to the development of new diseases until the inflammation is resolved. So in the case of inflammation indirectly induced by fascia dysfunction, so that bidirectional feedback loop between the slower lymph flow and the inflammation may lead to non-resolving or chronic inflammation. And thus, the fascia dysfunction may lead to development of carcinoma. So research has found that non-resolving inflammation can contribute to the development and survival of oncogenic cells by promoting angiogenesis, lymph angiogenesis, and even damaging the DNA of healthy cells. So angiogenesis and lymph angiogenesis are the development of new blood vessels and lymph vessels, respectively. And the growth of these new vessels may allow cancer cells to leave a specific area and travel to invade other parts of the body. And this is called metastasis. So cells and molecules in this process are crucial in the inflammatory response, but many cells and molecules can create pathways that contribute to the development of cancer during chronic inflammation and even in acute inflammation. So cells such as macrophages, dendritic cells, and T lymphocytes may contribute to this oncogenic cell proliferation, or just the development of new oncogenic cells. So in addition, cytokines, which are a type of protein secreted by the immune system, can be produced by inflammatory cells and can induce oncogenic cell proliferation as well. So in addition, lastly, nitric oxide can convert endothelial cells, which are just normal cells in that area, to oncogenic cells in a process known as carcinogenesis. So here is a comprehensive list of some of the cytokines that may be released in the inflammatory environment. And all of these cytokines, which is not all the cytokines that are listed, but these ones specifically can have an impact on the development of carcinoma. So to recap, the densification or fibrosis of connective tissue may lead to inflammation, either directly or indirectly through the lymphatic system. So because inflammation can create a tumor-promoting environment, it can be inferred that the densification or fibrosis of connective tissue may lead to the development of carcinoma. And new research has also pointed towards this. In a recent study, it was found that women with dense breast tissue were 17 times more likely to have breast cancer, which is a form of a carcinoma, than women with healthy breast tissue. So not only does this research need to, not only does more research need to be done on this topic, but the densification and fibrosis of connective tissue needs to be studied in relation to almost all inflammatory diseases, as it might have an impact. So hopefully we can gain a better understanding of connective tissue in the near future, but can also apply this understanding towards new forms of treatment. Here are some of my references, and thank you. I would love to answer some questions.
Thank you, Aneesh. As we wait for questions to come in, I can start. What kinds of interventions are available for people? You know, you've laid out that dense tissue is a problem in general. Are there any steps people can take to reduce the density of this fascia?
Yeah, there are like many different steps. So one would just be moving around a lot more and being a more physically active person. So a lot of athletes would not have this problem. They might because of injuries or, like I said, surgeries, but just moving around more allows the tissue to become healthier as some load is being added onto it. And in turn, this creates, studies have shown this creates healthy fascia. And then also, while I was doing research on this topic, I found that even acupuncture actually helps fascia a lot. So I found that really interesting. The way that the needle interacts with the connective tissue really helps with fascia health.
That's super interesting. Okay, so Jin did put a question in the chat, but I think you addressed it in your talk, and I want to make sure we have enough time for Baran's talk. So if you could just respond to that question in the chat, that would be awesome. So I'll let Baran get on to his talk next.
I'm gonna share my screen. Is my screen visible?
Yes, it is.
Hi, I'm Baran Beydilli, and my research project was about the diversity and abundance of organisms in tide pools as a function of distance from the ocean, and I did this in Southern California. Some background and intro about my topic. So tide pools are shallow pools of water along the coast. They form in rocky areas, and they're formed by when water gets trapped in depressions in the rock as water recedes during low tide, with water depth of tide pools ranging from a few inches to a few feet in some of the bigger ones. They can also be formed through erosion by water moving in and out of an area and eroding the surrounding rock. They're the home of many organisms like hermit crabs, and other small species, sometimes even small fish. There have been some previous studies relating to tide pools, like how environmental factors affect tide pools, like freezing and wave exposure, but no studies have been done about how about the impact of the distance from the ocean of the tide pools to the community composition and diversity of species in the tide pools. And these are some pictures of tide pools formed by erosion in Southern California. So my objective was to test how distance from the ocean affects the diversity and abundance of organisms in tide pools. And my hypothesis was that the closer to the ocean, the more life there will be because the waves can transport the organisms to the tide pools easier, and there'll be more water and nutrients in the tide pools close to the ocean, which is the organism's natural habitat, so they'll prefer the close tide pools. And I decided to do this as my research topic because I've always been interested in biology and like how ecosystems function. So I thought this would be a good way to further my knowledge. And my methods and materials. So this experiment was done with very simple materials. The only things I needed were a 5 by 5 inch wooden frame as shown here and a tape measure. To do this experiment, I measured out 3 30-foot long transects, one close to the ocean as shown in the close transect here, one a medium distance away as shown in this transect, and one a far distance away as shown here. And obviously with my hypothesis being that they were close to the ocean, I started there and expected to see like a lot of organisms. And to do this experiment, I measured out each transect's length and width, and using those numbers, I randomly generated 30 different data points, 90 different data points for each transect. And I would place my, this slide. I would place my wooden frame at each of those data points and record what I observed in the frame. And here are my results for hermit crabs and crabs. So in each chart, there were 90 different data points, and on the y-axis, it's the hermit crabs and crab count per 25 square inches. And on the x-axis, it's each transect and the average amount. Overall, my results showed that the far third transect had the highest concentration and amount of hermit crabs compared to the middle transect and the closed transect. Hermit crabs were mainly found in large clumps when they were farther away from the ocean. And this is likely because they move into and clump up in whatever remaining water is left when the tide goes down. They clump up in these pools of water because marine hermit crabs can only live outside of the water for a short amount of time because they have to keep their gills damp to survive. And this mostly occurred in the far transect, which led to many data points in this transect having 0 hermit crabs and some having up to 26 hermit crabs within that small area with a mean of 3. This result is contrary to my hypothesis because I thought that organism population would decrease by distance from the ocean. However, there was significant variation in the data, and I used standard deviation in my graph as a measure of variation in the data as shown with these error bars. And for each transect, the standard deviation was larger than the average number of hermit crabs by transect, so the differences across the transects were not significant. The crabs chart was also similar with 90 data points and across the 3 transects, but crabs were like very few and far between. Most of them were found in the close transect, and a few were found in the middle transect, with I believe only 1 found in the far transect. Again, there is significant variation in the data, meaning that these differences are not significant. And I also did this experiment with relating to mussels and coral. These charts again have 90 data points each, and the mussels were mostly found in the close transect, with a few in the middle transect and none in the far transect. Similarly, coral was present mostly in the close transect and none were found in the far transect. Similar to the hermit crab data, there was no significant variation in data for crabs, mussels, and coral as indicated by large standard deviation error bars. Therefore, the observed differences across the transects are not significant. While the difference in terms of quantity of organisms across transects may not be statistically significant, results still suggest there is more diversity in tide pools closer to the ocean, as there were other non-focal organisms, such as sea anemones and sea urchins found in those close tide pools, with the far ones containing none of these other organisms. The far tide pools had the least variety of organisms. However, it had a high concentration of hermit crabs and had more hermit crabs than the other transects. This is likely because the tide pools across the close transect had more nutrients and were flooded more with fresh ocean water than those across the middle and far transects. Conclusion and some future steps. So my results were consistent with my hypothesis for my crabs, mussels, and coral, where the average number and presence of these organisms decreased by distance from the ocean. But for hermit crabs, they were actually against my hypothesis because they were mostly found in the far transect. In general, the tide pools close to the ocean had more diversity, and this was likely due to these tide pools having more nutrients to support a more diverse organism because they were more frequently flushed with fresh water from the ocean to get crucial nutrients for these creatures to survive. And some future steps, this study can be expanded with additional data points because with only 90 data points, it was harder to get a more accurate result. And I think that with more data points, the results would be statistically significant and the standard deviation would be, error bars would be less than the averages. And you could see a difference in these organisms across the tide pools. And some other steps for this could be the research conducted in different tide pools with different geographical factors such as distance from cities, different topography, climate, other environmental factors, and how this would affect the experiment, and seeing how that could affect the community composition in the tide pools. I want to thank my mentor, Christian Gray, for the help in doing this research and give me advice along the way. And I'm open to any questions.
Thank you so much, Baran. So sorry for getting your name wrong. We'll do better. Samantha has her hand raised, so go ahead.
So I live right by a coastal environment by Crystal Cove, and I know that depending on what the shifting tides are, certain parts of the tide pools are exposed at certain times. So I don't know if you're able to answer this question, but for the species that are living on the parts of the tide pool that are farther away from the ocean, how are they able to get like the nutrients or maybe like water exposure that they need when they may be unexposed to like the seawater for a prolonged amount of time?
So that's why there weren't like too many organisms in that one, because the longer tides, the high tides would flush in nutrients, but they would the nutrients would go away obviously when the tide goes back down, leaving them in the close transects. And so that's why hermit crabs, that's why we found them like clumping up in these like small water pools in whatever was left there. And other species like coral and mussels just couldn't survive in those environments, so none were found there. And the crabs, they can like move around a little, and so they would stay like closer to where the nutrients were and in like a better environment in the close transects.
Thank you.
Yeah.
I have a quick question. Thank you so much for that presentation. My question is what was the experience like in terms of setting up these experiments when obviously your mentor was virtual and you had to go do it yourself? Can you tell us a little bit about what was challenging about that and how you overcame those challenges?
Well, I would discuss with my mentor and like take pictures of the environment and discuss like possible experiments, and we work together to figure it out. Honestly, the most difficult part of doing the experiment was probably getting to the tide pools, because the low tide, these tide pools are like along the coast, and it took a while to walk there across the rocks, and the low tide was only there for like a certain amount of time. So it was kind of challenging to get the data and then get out before you get stuck there.
Got it. Very cool. Thanks for sharing.
Yeah.
Would suggest a sponsored trip to the North Shore of Oahu to get more data points. Maybe you can put that on your plans. A sponsored trip to the North Shore of Oahu.
Oh, that would
get you a lot more data points. So I hope you continue the research. Did you find, were there any other organisms in some of those tide pools that that you didn't document? Fish species or anything like that?
Yeah, so I didn't do much documentation about sea urchins and anemones due to for the sea urchins, we only found them like a few days out of the different times I went there. So it was kind of hard to get consistent data on those. And with the sea anemones, there was like too few to really get good data on. So yeah, there were those two that I didn't collect data on.
Got it. Cool.
I had a question about the nutrients. You mentioned a couple times that the closer transects might be getting like more nutrients in their water. Have you considered testing? I'm thinking of like testing a pool for its pH and ammonia. Have you considered like testing for your chemicals in the water that way and seeing any differences between the closer and farther tide pools like that?
I hadn't considered that, but that's actually a really good suggestion. And I think that could like lead to some better conclusions from the experiment to see what really is causing this.
Awesome. Okay, well, we're definitely close to time. We can wait for any more questions, but seeing none, I'm happy to let everyone go back into the lobby. Thank you for, oh, go ahead.
I had a question for Aneesh that I didn't get a chance to ask, which is, what are some of the original causes of, or external causes of inflammation and fibrosis? Are there like external events or anything like that would be the initial cause of some of the inflammation?
So for fibrosis, fibrosis can happen, like I said, from maybe injuries or limited activity, things like that. However, inflammation can be caused from like almost like a multitude of things. It could be from like injuries, like I said before, but then also it can be from diseases, like if you some pathogens invade your body, which is like maybe bacteria cells. Your body also has to send an immune response for that too. And anytime your body has to send an immune response, inflammation will occur. So it could be for that. It could be more like I said, injuries. It could be like many different things.
Got it. Got it. All right. Thank you.
Yeah.
Wonderful. All right, well, with a minute left, I'm happy to say thank you to all of our wonderful speakers. It was really great seeing all of your presentations today, and have fun at the rest of the symposium. Bye.